Photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Have we ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. Green plants carry out photosynthesis, a physiochemical process by which they use light energy to derive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all foods on Earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform light energy into chemical energy. What do we know? Let us try to find out we have already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, green pigment of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black pepper and one that was exposed to light. On testing these leaves for starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Another experiment you may have carried out is the half-leaf experiment, where a part of leaf is enclosed in a test tube containing some KOH-soaked cotton, which absorbs CO2, while the other half is exposed to air. This setup is then placed in light for some time. On testing for starch later in the two halves of the leaf, you must have found that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative. This showed that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley, 1733 to 1804, in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar soon gets extinguished. Figure 13.1 A, B, C, D. Similarly, a mouse will soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed in the air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he will need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Ingenhousz showed that the sunlight is essential to the plant processes that somehow purifies the air followed by burning candles or breathing animals. Ingenhousz is an elegant experiment with an aquatic plant showed that in bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is the only green part of the plant that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as the starch. His later studies showed that the green substance in plants, chlorophyll as we know it now, as located in special bodies later called chloroplast within plant cells, he found that the green parts in a plant is where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann and that illuminated a green alga cladophora placed in suspension of aerobic bacteria. The bacteria were used to detect the sites of photo evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B, discussed in section 13.4. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as CO2 plus H2O, light CH2O plus O2, where CH2O represented a carbohydrate, a glucose, example, a glucose 6 carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by microbiologist Cornelius Van Niel 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by 2H2A plus CO2 light 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis, where H2S instead is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending on the organism and not O2. Hence, he inferred that the O2 evolved by the green plant comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that will represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O gives the reaction in the presence of light C6H12O6 plus 6H2O plus 6O2, where C6H12O6 represents glucose. The O2 release is from water. This was proved using radioisotope techniques. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as a substrates are used in the equation given below? Where does photosynthesis take place? You will of course answer in the green leaf, or you may add in the chloroplast. Based on what you earlier read in chapter 8, you are definitely right. Photosynthesis does take place in the green leaves of plant, but it does so also in other green parts of the plant. Can you name some other parts where you think photosynthesis may occur? You will recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Using the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is the membranous system consisting of a grana, the stroma lamella, and the fluid stroma. There is a clear division of a labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In a stroma, enzymatic reactions incorporate CO2 into the plant, leading to the synthesis of sugar, which in turn forms starch. The former set of reactions, since they are directly light driven, are called light reaction. The latter are not directly light driven but are dependent on the product of light reaction, ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reaction. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. How many pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves? Even in the same plant, we can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigment shows that the color that we see in leaves is not due to a single pigment but due to four pigments. Chlorophyll A, bright or blue green in the chromatogram. Chlorophyll B, yellow green color. xanthophylls yellow color, and carotenoids yellow to yellow orange color. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A pigment to absorb lights of different wavelengths. Of course, you are familiar with the wavelengths of the visible spectrum of light as well as the waveguide. From figure 13.13a, can you determine the wavelength color of light at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelengths too? If yes, which one? Now look at the figure 13.3b showing the wavelengths at which maximum photosynthesis occur in a plant. Can you see that the wavelengths at which there is a maximum absorption by chlorophyll A, that is in the blue and the red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.3c, can you see that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together shows that the most of the photosynthesis take place in the blue and the red regions of the spectrum. Some photosynthesis does take place at the other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophylls is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelengths of incoming light to be utilized for photosynthesis but also protect chlorophyll A from photooxidation. Light reactions of the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH. Several complexes are involved in the processes. The pigments are organized into two discrete photochemical light harvesting complexes LSC within the photosystem 1 PS1 and photosystem 2 PS2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LSC are made up of hundreds of pigments molecules bound to proteins. Each photosynthesis has all the pigments except one molecule of chlorophyll A, forming a light harvesting system also called antennae. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecules form photosystem in PS1. The reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in PS2 it has absorption maxima at 689 nanometer and is called P680. The electron transport. In photosystem second, the reaction center chlorophyll A absorbs 680 nanometer and the wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor which passes them to an electron transport system consisting of cytochromes. This movement of electrons is downhill in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosystem PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a great redox potential. These electrons then are moved downhill again, this time to a molecule of energy rich NADP plus. The addition of these electrons reduces NADP plus to NADPH plus H plus. This whole scheme of transfer of electrons starting from the PS second uphill to the up acceptor, down the electron transport chain to PS first, excitations of electron transfer of N to another acceptor, and finally downhill to NADP plus, causing it to be reduced to NADPH plus H plus, is called the Z scheme due to its characteristic shape. This shape is formed when all the electron carriers are placed in a sequence on a redox potential scheme. Splitting of water. You will then ask, how does PS second supply electrons continuously? The electrons that were moved from photosystem second must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS second. Water is split into H plus, nascent oxygen and electrons. This creates oxygens, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. We need to emphasize here that the water splitting complex is associated with the PS second, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and the O2 are found likely to be released in the lumen or on the outer side of the membrane. Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bond. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS second and then the PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus are synthesized by this kind of electron flow. When only PS first is functional, the electron is circulated within the photosystem 1 and the phosphorylation occurs due to cyclic flow of electron. A possible location where this could be happening in the stroma lamellae, while the membrane or lamellae of the grana have both PS1 and PS2 of the stroma lamellae membrane lack PS second and as well as NADP reductase enzyme. The excited electron does not pass on to NADP plus but is cycled back to the PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of NADPH plus. The cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nanometer are always for excitation. Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism like in respiration and photosynthesis too. ATP synthesis is linked to development of a proton gradient across a membrane. This type these are membrane of the thylakoid. There is one difference row here. The proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria where electrons move through the ETS. Let us understand what causes the proton gradient across the membrane. We need to consider again the processes that take place during the activations of electrons and their transport to determine the steps that cause a proton gradient to develop. Since splitting of the water molecule takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. As electrons move through the photosynthesis, protons are transported across the membrane. This happens because the primary acceptor of the electron, which is located towards the outer side of the membrane, transfers its electrons not to an electron carrier but to an H carrier. Hence, this mobile removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the NM side of the lumen side of the membrane. The NADP reductase enzyme is located on the stroma side of the membrane along with the electrons that come from the acceptor of electrons of PS1. Protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number while in the lumen there is accumulation of proteins. This creates a proton gradient across the thylakoid membrane as well as measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of the gradient that leads to release of energy. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the F0 of the ATPase. The ATPase enzyme consists of two parts. One called the F0 is embedded in the membrane and forms the transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called the F1 and the protrudes on the other surface of the thylakoid membrane on the side that faces the stroma. That breakdown of the gradients provides enough energy to cause a conformational change in the F1 particle of the ATPase, which makes the enzyme synthesis is several molecules of energy-packed ATP. Chemiosmosis requires a membrane and proton pump and proton gradient and ATPase. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATP has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATPase enzyme that catalyzes the formation of ATP. Along with the ATP, NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma responsible for fixing CO2 and synthesis of sugar.